Alright, so in this video we're going to talk about some of the accessory organs of the digestive system. So we'll be looking at the pancreas, the liver, and the gallbladder. So here we go. Pancreas are a gland that have both an endocrine and an exocrine function. We haven't talked much about the endocrine function, although we have had some conversations about its role in regulating blood glucose by the production of insulin and glucagon. What we're going to talk about most in this chapter, though, is its exocrine function and the pancreatic juices that it secretes. Pancreatic juice is a combination of enzymes that are responsible for chemical digestion and a very high concentration of bicarbonate ions that work to neutralize the acids that are produced in the stomach and mixed into the chyme, and also inactivate the pepsin that has been produced in the stomach. So, pancreatic juices are produced by a set of cells that are called the pancreatic acinar cells, and they produce secretions that are released into the pancreatic duct, and the pancreatic duct then delivers these secretions to the duodenum, which is the first 10 to 12 inches of the small intestine. So the secretions include an enzyme called pancreatic amylase, which, just like salivary amylase, is responsible for breaking down starch and glycogen into disaccharides that can be absorbed. There's also pancreatic lipase. So if it's lipase, what is it breaking down? Well, it's breaking down the triglycerides, the dietary fats that we consume, and it strips off the three fatty acids from the glycerol backbone, producing the fatty acids that are released and monoglycerides. So we take away just two of the. So if you remember, this is triglyceride, hydrogen, and then an O that has the fatty acid attached. So we can strip away, we can break this bond and break this bond and take off the two fatty acids, and then what we're left with is a glycerol backbone with a single fatty acid attached to it, which is what a monoglyceride is. Pancreatic juice also includes a type of enzyme called nucleases. These break down the nucleic acids, and recall, nucleic acids are DNA and RNA that we find inside the cells, and it breaks them down into their constituent nucleotides. So it's the nucleotides that are the monomers for a nucleic acid polymer. And then finally, there are a number of proteases that we find in pancreatic juices as well, that are responsible for breaking proteins down into short chains of amino acids called peptides. So a peptide, that's a short chain of amino acids. We usually think of a peptide as having less than 100 amino acids attached to it. Proteins are 100, if you see approximately 100 or more amino acids, that's when we start talking about proteins. So like pepsin in the stomach, the proteases are secreted in inactive forms, and then they are activated by other enzymes to make the inactive form active. So we produce trypsinogen, and trypsinogen is converted in the small intestine into trypsin, And then trypsin helps to activate chymotrypsin and carboxypeptidase. And then these three enzymes go about doing their work to digest the proteins that we have eaten. So to help you remember which of the names are involved with breaking down proteins, the thing to remember is that, remember, pepsin is the fourth protease. If it has trip, like trypsin or chymotrypsin, or if it has pep, carboxypeptidase, pepsin and carboxypeptidase, then we know that that's an enzyme that's responsible for breaking down proteins. So trip and pep are the protein enzymes. So how is it that the pancreas knows that it's time to release its secretions? Well, when acids enter the When acids enter the duodenum, that results in the release of a hormone called secretin, or secretin. It's probably the more appropriate way to say it. Secretin signals back to the pancreas that we need to have bicarbonate ions released. So in response to secretin, bicarbonate ions are released from the pancreas. The other regulator of pancreatic secretion is provided by the hormone cholecystokinin. I've already talked a little about cholecystokinin. It also stimulates the release of pancreatic enzymes. So several functions then for cholecystokinin. Remember, it was cholecystokinin that decreases the hormone, the movements of the stomach, the smooth muscle of the stomach. It also stimulates the secretion of pancreatic enzymes. So secretin and cholecystokinin are responsible for regulating pancreatic secretion. Okay, so that was the pancreas. Now let's talk a little about the liver. And if you turn to page 671, you can see a little bit about how the liver is organized in that picture there. So figure 1727 and figure 1726. Both of those are going to be useful for this conversation. So the liver is enclosed in a fibrous capsule. So I'm thinking, we've talked about capsules before. When we talked about lymph nodes, remember lymph nodes were enclosed in a fibrous capsule. So this is connective tissue. It's a dense, irregular connective tissue that helps to protect the surface of the liver, because the liver is a fairly delicate organ. If you've ever cooked with liver, you know that it can be squeezed. It's kind of springy consistency, so relatively delicate. It's separated into 4 relatively large lobes by folds of visceral peritoneum. So the round ligament. The visceral peritoneum that covers that is external to this fibrous capsule dives into the liver structure, and it separates the liver into the 4 lobes. There's a really large right lobe separated by the left lobe by the falciform ligament. So right and left are separated by the falciform ligament. And then there are 2 smaller lobes, the caudate lobe and the quadrate lobe. The lobes are built of structures called hepatic lobules. These are liver cells that all surround a central vein. And the veins enter into the blood supply in the liver, is formed by very dense networks of capillaries, and the blood is supplied by the hepatic portal vein, which brings all of the blood from the small intestines into the liver, where capillary, they diverge, forming the capillary networks of the liver, and so the liver gets the first shot at all the digested nutrients that are absorbed into the blood there around the small intestines. So if you remember, we talked about portal systems a little bit already. So a portal vein is a vein that is in between two capillary networks. Normally we think about blood flow coming through an artery to an arterial to a capillary network, then into venules and into veins, and then back to the heart to be recirculated. Well, in this case, though, in the case of portal networks and portal veins, we go through a capillary network. Venules merge to form veins. The veins then will subdivide to form venules again, and then form another set of capillary networks. So you go to a second capillary network prior to making it back to the heart. So the capillaries of the small intestine lead then not back to the heart, but first to the capillary networks that are found within the liver. And in between those two capillary networks, we form large blood vessels called the hepatic portal vein. So what is it that the liver does? It is responsible for the most important, or the largest function would be its responsibility for metabolic regulation of the body. So when we talk about metabolic regulation, what I'm talking about is it's the liver that determines what nutrients are going to be released out to the body to be used by the body. So the liver is constantly paying attention to the blood that is passing through, and it recognizes when there are deficiencies in specific nutrients, and it can release those nutrients that it has stored in the cells there of the liver. So hepatocytes are the liver cells, and their job is to adjust the levels of circulating nutrients before we send the blood out into the general circulation. And the hepatocytes are capable of both breaking down and synthesizing carbohydrates, lipids, and proteins. And included in this function, of course, is the production of all the plasma proteins that we talked about back in the blood, in the chapter on the blood, that are important to regulating osmotic pressure of the blood, the fibrinogens that we have circulating around dissolved, waiting to coagulate. All of those plasma proteins are produced by the liver. So when a body is low on any one nutrient, it can use other nutrients as fuel or as feedstock to create the nutrients that we are low on. Most of the nutrients we require can be synthesized by the hepatocytes of the liver, with a few exceptions, of course, especially the essential amino acids, the amino acids that we have to eat in order to have them in our body. The liver also responds to the hormones insulin and glucagon. In the case of insulin, then, it stores glucose as glycogen. In the case of glucagon release, then it releases the glycogen, it breaks up the glycogen and releases glucose that was stored previously. So metabolic regulation is a really important function of the liver, but there are other functions as well. It stores materials, especially glycogen. It stores iron that has been recycled from blood cells that were recycled, broken down, And it also stores a lot of vitamins, and so the liver is a rich source of vitamins A, D, and B12, especially if you eat livers of animals. It also filters blood in the same way that the spleen does some blood filtration. Well, the liver does the same thing, and it can remove damaged red blood cells, cellular debris, and pathogens, because there are resident lymphocytes that take up their home or make their home in the liver. It can do detoxification, and this is because liver hepatocytes have relatively large, smooth endoplasmic reticulum, or reticula, I guess. So each of the cells have a large collection of smooth ER, and that smooth ER is the surface on which many enzymes are found that are responsible for detoxifying the toxins that are delivered to the liver by digestive system. It also removes pathogens or the toxins produced by pathogens. And then, last of all, is the function of producing bile. Bile is a fluid that it makes and delivers then to the duodenum via the bile ducts. So here's an exocrine function, again, of the liver cells. Bile is composed of bile salts, bile pigments, cholesterol, and electrolytes. The function of the bile is to emulsify fats. Emulsify fats. What in the heck does that mean? Fats in water tend to form large globules. Imagine salad dressing made of oil and vinegar that you have shaken up. So you make your own homemade salad dressing. You give it a good shake, and the fat will break into what appear to us to be small little globules, but what are in actuality relatively large globules that are then very difficult for enzymes to be able to go and attack. So if you imagine these are the enzymes, and they've got only a short time, maybe an hour or two, as the food is residing in the duodenum, to really do their work on it. Excuse me. So that's a fairly inefficient way. But if we could somehow break this fat globule up into a bunch of smaller fat globules, like this, then when the enzymes come to attack it, they'll actually have time to digest all the triglycerides in those fat globules as they do their work. So the same number of enzymes can get a lot more work done if we emulsify the fat. If we take the large fat globules and break it up into really tiny fat globules. So if you buy a creamy salad dressing, that is a salad dressing then to which an emulsifying agent, often lecithin, has been added to help ensure that the fat remains in tiny little globules rather than reforming into large globules that then float to the surface of the salad dressing. So the bile salts, their job is to emulsify those fats, and that allows the pancreatic lipase to more easily interact with and digest those fats, and it enhances later on, bile salts do, it enhances the absorption of fatty acids, cholesterol, and the vitamins that are dissolved in the fat. The bile pigments are actually a waste material. So as the bile is being squirted into the duodenum, we are excreting bile pigments. Bilirubin and biliverdin are the two bile pigments that we get rid of as we go to break down hemoglobin that's found in the red blood cells. All right. That leads us then to the gallbladder, and here is a dissected gallbladder. We'll talk about that in just a minute. Pretty horrific looking sight. The gallbladder is a small not duct, but a side chute off of the bile duct. So if you think about the bile duct heading down from the liver to the duodenum, here's the small intestine. Then here, off of a little side of the bile duct, we find the gallbladder. So there's a sphincter muscle here that controls the flow of bile into the duodenum, and so it's not open all the time. And so, but the liver is constantly dripping down the bile that it's making, and so the bile duct will fill, and when it gets up to the level of the duct that leads into the gallbladder, then the overflow fills up into the gallbladder as well, and then keeps filling. So when we then finally do use the bile that's accumulated, we release it in. That draws down the bile and allows for more bile to leak out, or to be squeezed out into the bile duct to deliver it out to the duodenum. So we make way more bile than we can actually use, and then what is secreted into the duodenum. And so a second job, not only of the gallbladder, is not only to store it, but a second job is to concentrate that excess bile. So it pulls out water from the bile that it's holding there in the gallbladder. Now, as we concentrate the bile, then the cholesterol and other salts that are found in the bile as they become more concentrated, it makes it easier for them to crystallize and come out of solution, so that they're no longer dissolved, but instead will accrete, collect, and form gallstones that then can sometimes block the flow of bile out of the gallbladder. So when a person passing a gallstone, if you can imagine trying to pass a relatively large stone like that out of the gallbladder, it's not possible. The smaller stones they may pass, but man, they cause a lot of pain as they are passed through the bile duct and into the duodenum. Cholecystokinin that is being produced that slows the activity of the stomach and encourages the release of the pancreatic juices that are being secreted by the pancreas, also stimulates the smooth muscle of the gallbladder to contract and deliver bile down to the duodenum. So cholecystokinin shows up with a third function now. So be sure to keep track of all the functions that we can attribute to cholecystokinin. All right, so there we have it. Everything you ever wanted to know about the accessory organs of the digestive system. And when we get together again for the next video, then we'll be talking about this